Well, thank you very much for that introduction, and I'm pleased and honored to be here today to address you at the outset of what will surely be an exciting adventure for this new Max Planck Institute. I want to credit Vahid for inviting me. We met when he visited Berkeley for a sabbatical last year, and we talked about his research and his vision for this new institute, and then he invited me to come and give this lecture, so I'm pleased to do that. So I'm going to talk about work in my laboratory for the past 10 years or so. My research has focused on how cells traffic proteins and membranes. For many years it was to study the mechanism of traffic of vesicles within cells that are involved in the process of protein secretion. But about a dozen years ago, I turned my attention to a topic of broad interest in not only cell biology, but in medicine, and that is the production of extracellular vesicles by cells. Now, all cells, even microbial cells, produce vesicles that are spread into the environment. At the extreme, we do so, and you find these vesicles in various sizes and composition in all of the bodily fluids that we produce. There are billions of them floating in the bloodstream. They are sentinels of the activity of all of the individual cells that are involved in producing them. Tumors, in particular, are prolific in the production of extracellular vesicles. And so I'll speak now and perhaps later in the discussion about the tremendous potential of using extracellular vesicles in liquid biopsy to evaluate early diagnosis and progression of cancer. But this talk is going to be purely about basic science, so let me go to the first slide. This is a cartoon of the two processes that are involved in the production of extracellular vesicles. There is a process of budding from the plasma membrane by analogy to enveloped RNA viruses such as HIV that bud from the cell surface. Whoopsie. That's not a vesicle, that's me. Okay. There's another process that produces a specialized population of vesicles called exosomes that are the byproduct of a process of endocytosis and internalization into vesicles within the interior of an endosome, maturing into a structure called the multivesicular body, which delivers its content for degradation in the lysosome. But it also, on occasion, is exposed to the cell surface where a bolus of these extracellular vesicles are produced. Now, these extracellular vesicles are characterized by a typical lipid bilayer. They have certain membrane proteins, some of which are characteristic of the origin, the tissue origin of their production. They have soluble interior luminal proteins, but interestingly, unlike vesicles that traffic within the cell, these extracellular vesicles carry small RNA molecules. MicroRNA is a particular interest in my lab because we've been able to show that certain microRNAs are very selectively packaged into these vesicles. For what purpose, that remains unclear. Now, the big question that we've sought to address, and which I will show you experiments concerning today, are what are the forces that generate these vesicles? How is their production controlled? And then secondly, perhaps even more importantly, what is the purpose? Why do cells bother to make these vesicles? Is there some purpose to them? They are widely believed in the literature by many people around the world to convey information in the form of signaling or direction of cargo molecules to other cells, to inform them and to drive different processes of differentiation or changes in cell metabolism. But I'm going to challenge that view with the two broad results that I'm going to discuss today. The first concerns forces that are involved in the production of these vesicles. First, let me just briefly describe the simple procedures that we use to fractionate and have defined two broad populations of vesicles, in this case produced and secreted by a breast cancer cell line, MB231. If you simply take conditioned medium from these cells and you centrifuge at high speed and collect the pellet and expose it to a buoyant density gradient, after a period of centrifugation, you can define two categories of vesicles. one of low buoyant density that is marked by certain proteins, some uniquely characteristic of these low buoyant density vesicles. These, we believe, are vesicles that bud from the plasma membrane. We have markers in these vesicles that are characteristic of the plasma membrane. At a higher buoyant density, we find a population marked very specifically by a protein called CD63, about which I'll have more to say later. And we've shown by isolating and characterizing these vesicles that they enrich a population, a small number of certain microRNAs, depending on the cell of origin, that are characteristically produced by these cells and which can be used to distinguish different kinds of cells. Now, let me go back. People have often suggested in the literature that the microRNAs that are packaged into these vesicles are processed, are matured by enzymes that are housed within the vesicles themselves. These are enzymes such as dicer or argonaute. But we found that these enzymes and proteins can be readily separated. They are not actually within vesicles. They sediment to the bottom of a buoyant density gradient. They are not buoyant. And so we feel that the microRNAs have been matured and then packaged into a vesicle that buds into the interior of the endosome. Now, in designing experiments to test the conditions that promote the production of these vesicles, two of my graduate students, Justin Williams and Jordan No, devised a really simple assay that we can apply almost in real time as cells are growing in culture. That's shown in the next slide. The assay involves the use of a membrane impermeable inhibitor of an enzyme luciferase. In this case, the luciferase has been introduced to the C-terminus of the coding, 3' end of the coding region of the gene for CD63. And the prediction is, according to the topology of CD63, that the luciferase enzyme would be contained within the interior of a vesicle that has budded from the cell surface or that has been produced by the exosome pathway. So Justin and Jordan found that if they took culture conditioned medium, they find luciferase, but sometimes they find it in fragments. Very often, though, the vesicles are intact. And so in order to follow the intact vesicle specifically, they designed an assay that included a membrane impermeable inhibitor of luciferase and a membrane permeable substrate. The impermeable inhibitor blocks the activity of anything that is in membrane fragments. The membrane permeable substrate is thus accessible and then detects the enclosed vesicles. You can see that a fraction of the material in the conditioned medium is susceptible to inhibition by the membrane impermeable inhibitor, whereas a substantial fraction remains intact unless the sample is incubated in the presence of detergent. So applying this assay with these two molecules, you can see a linear time course of production of extracellular vesicles as diagnosed here. And since we're marking CD63, we're in this case looking specifically at exosomes, and this will become important in a moment. Now, I'm going to summarize several broad experiments that reach the conclusion that conditions that damage cells, such as often occur in physiologic conditions where cells are stressed. These are conditions that we were able to impose in cell culture. Conditions that damage the plasma membrane, conditions that elicit a repair process, also produce an abundant secretion of exosomes. And that data is shown in this slide. So here are the conditions that Justin and Jordan used. The first is simply to treat cells with ionomycin, which permeabilizes the plasma membrane to calcium, which flows into the cell. And this is known to elicit a membrane repair process. And at the same time as this process occurs, they observed about a 30 to 40-fold increase in the secretion of intact exosomes compared to a control sample, quite a dramatic change. Another more drastic condition was to treat cells with a toxin, streptolysin O, that pokes holes in the plasma membrane. Under these conditions, in the presence of extracellular calcium, a 20-fold increase in the secretion of exosomes is observed. And more mild conditions, such as simply stressing cells by pipetting them repeatedly through a narrow gauge needle without actually rupturing the cells, that stress, which is at a level that's often encountered in many tissues in our body, was enough also to elicit in even low regime or in high regime, a twofold increase in the secretion of these exosomes. Now, if the exosomes so produced are resolved on a buoyant density gradient, they sediment to a high buoyant density position characteristic of exosomes and of CD63 as detected independently. Now, the process of plasma membrane repair is not a new phenomenon among experimental biologists. It was observed already over 100 years ago by classical developmental biologists, as we'll see in this experiment shown from a publication from over 100 years ago. If you treat sea urchin eggs under rupturing conditions, you can see a streaming of cytoplasm out of these cells, and visualized alternatively, you can see by poking a needle into the plasma membrane a burst of cytoplasmic content followed within minutes by closure of the rupture, to the point where this ruptured egg can actually go on, be fertilized, and develop into a sea urchin. So there's an enormous process of damage repair. This has been studied over many years, principally by an investigator in the US by the name of Norma Andrews. She and her colleagues now at the University of Maryland have published beautiful work showing that when the plasma membrane is damaged, the damage is patched by the recruitment of lysosomes, which migrate to the cell surface, fuse at the point of damage, and then release their soluble lysosomal content to the cell exterior. At the same time that happens, the lysosomal membrane becomes integrated into the plasma membrane of the cell. That membrane can eventually be internalized by endocytosis and recycled, thus restoring the cell to perfect viability. Now, we've done this by not just poking needles into a cell, but let's see if we can get this movie going. Yes, so these are cells that have had all their membranes labeled with a fluorescent dye, and the point of the arrow is where a laser damage was introduced at the cell surface. Let's see if I can do this over again. Oops, go back. No, I guess not. Anyway, you saw briefly membrane flowing from the point of damage to the cell exterior, with the cell being repaired and retaining viability within a couple of minutes. Now, we asked whether the conditions that allow this repair, which, as I mentioned, the Andrews lab has shown invokes the lysosome for the repair process, may also mobilize the multivesicular body into which exosomes have been introduced by a budding process. It's known that the multivesicular body is very similar to the lysosome in terms of its composition. They both have the marker protein CD63 and other proteins that are characteristic of the endolysosomal pathway. So we asked a simple question: can we demonstrate that the process that invokes the lysosome may also employ the multivesicular body and thus account for the enormous burst of secretion of exosomes that I showed you a few slides ago? We did this in an experiment that Jordan did to ask whether one could see cytoplasmic proteins recruited to the multivesicular body in an in vitro binding assay, and to assess whether the same proteins that are known to be mobilized for lysosome recruitment do so for the multivesicular body. So here's the experiment. Jordan took cells that had the CD63 protein marked with an epitope that allowed him to immobilize endosomes and lysosomes onto beads to which an antibody had been attached. These beads can be mixed under conditions where it's just beads alone with a crude broken preparation of cells, or the beads can be, with the antibody coated, can be mixed in the presence or in the absence of calcium. Calcium here, and on addition of EGTA to block the calcium on the right. And these samples then are sedimented so that the beads simply collect everything that has bound. We could show that a lysosomal and endosomal membrane protein, LAMP1, is recruited under both conditions, but is not recruited when the beads lack antibody. In the next experiment, the sample that was incubated in the presence of calcium, the beads were then collected and washed with EGTA so as to release those proteins dependent on calcium that had adhered to the beads in the first place. And the results of that are shown in the panel on the right. In the middle is this experimental lane, where it's evident that proteins, a number of proteins that are boxed in red, have been recruited that are not recruited if the incubation had been conducted in the absence of calcium. These proteins were removed, excised, and sequenced, and Justin and Jordan found a subset of the very same proteins that Norma Andrews and her colleagues had described. They are the annexins, calcium-binding proteins. There are a number of genes that encode proteins called annexins, 1 through 6, and another protein called copine-3, and I'll show you the results of just 2 of those in the next slide. So in this experiment, what Jordan did was to use shRNA to ablate the expression with 2 different shRNA constructs of either annexin A6 or of copine-3. And by immunoblot, it's evident that his conditions of depletion were quite rigorous. The protein is reduced at least 80%. And then the cells from which these 2 proteins had been independently reduced were evaluated by the treatments that I've shown you before to mobilize exosomes for secretion. Under basal conditions, these cells produce a low level of exosomes as detected during the course of this incubation. Whereas those cells that had annexin A6 or copine-3 reduced by the level shown here, similarly reduce quite dramatically the exosomes that are secreted in the presence of ionomycin during just a brief 30-minute incubation. So we conclude from this set of experiments that these 2 proteins, likely more annexins, are required at least for the secretion of exosomes, and we believe that is as a result of the mobilization of multivesicular bodies to the cell surface. So let me then just summarize this part of the story before I tell you about the other pool of vesicles that are released in even more dramatic fashion. So we have a simple model where damage to the plasma membrane by any of a number of different physical, chemical, or toxins, calcium rushes into the cytoplasm that mobilizes calcium-binding proteins to the multivesicular body, which are then delivered to the point of damage by membrane fusion of the endosomal membrane with the consequent release of exosomes in a burst. Now, we believe that this is going on all the time under normal conditions, but we also wondered whether the conditions that I've just described apply to the production of vesicles that are budding from the plasma membrane, not involving the mobilization of the multivesicular body. So what we did was, under these conditions of elicited secretion, I showed you that it's possible to resolve the 2 broad populations of exosomes and budded vesicles on a buoyant density gradient. So what Justin did was he did this under conditions that allowed quantitative mass spec analysis of the proteins that sediment at these 2 positions on a buoyant density gradient. And he was able to show that the proteins that sediment along with vesicles at low buoyant density were enriched in a number of these annexin proteins. These are proteins that are inside of the vesicle. They are not proteins that are present on the cell surface. In contrast, the vesicles that sediment to high buoyant density, such as CD63 and others, are shown here. There is a really very clear distinction in the composition of these 2 broad populations of vesicles. So given this, it became interesting to examine independently if one of the annexins is itself also packaged during the very conditions that elicit this burst of secretion, as I showed you for exosomes. So we can visualize this. Let's see if I can get this to work. This movie, yes, so follow the arrow. This now is annexin that is labeled with M scarlet, and a laser ablation of the cell surface shows a dramatic cap that forms at the point of damage. And if you follow this closely, you see debris that's spread from the cell very actively during the first 30 seconds or so of this incubation, followed by a complete repair of the patch after just 1 or 2 minutes. So this is an indication that an exon is being packaged into what we think is the low buoyant density population quite actively during just a minute after damage is introduced. In fact, it's possible to separate, as I've indicated previously, these two populations of vesicles in this experiment. The high buoyant density population that sediments to a position in fractions 10 and 11 of this density gradient is marked by CD63, whereas the lower buoyant density vesicles are marked by their appearance here, and these are, in this case, annexins 1 and 2. It's possible also to take the high buoyant density vesicles and subject the isolated vesicles to conditions of immune precipitation using an epitope-tagged form of CD63 at the surface of the vesicle with an antibody bringing it down on a bead. Justin was able to show that essentially none of the annexins sediment along with those vesicles that have CD63. These are the ones that have been eluted or have been centrifuged under these conditions. So there's really two very distinct populations of vesicles. Now, it was possible then to compare directly the efficiency of production of annexin-containing vesicles and CD63-containing vesicles using the trick that I introduced in one of my first slides, which is shown here. This now is the production of CD63 N-luciferase vesicles, shown in the brown squares, during the first minutes of treatment with the pore-forming protein streptolysin O, a quite remarkable increase in the secretion of annexin-containing vesicles, distinct vesicles produced under these conditions, and dependent on incubations with this streptolysin O that are sublytic. These are not toxic levels. The cells can repair themselves quite readily. So we see under these conditions quite dramatic evidence for the production of two broad categories of vesicles under conditions that we believe mimic those that occur under normal circumstances. Indeed, I think my next slide will show we've looked at the forces that may apply in all the various tissues, some of which have been measured, the levels of stress have been measured by fluid shear in the major arteries. The level of shear that cells experience under these conditions are comparable to what we were able to use to show that there was at least a twofold increase in the secretion of exosomes. We know that platelets involved in blood clotting as a result of the physical association with this process also elicit a tremendous increase in the secretion of both populations of vesicles. And obviously, the act of muscle contraction likewise stimulates, in other cases, the secretion, in other evidence, the secretion of vesicles. Indeed, it's been shown in human physiology experiments that after a period of vigorous exercise, the level of CD63 in the bloodstream increases quite dramatically. So we conclude from these and other experiments that exosomes and, in general, extracellular vesicles may being produced by normal physiologic processes as a consequence of the need to repair damaged plasma membrane, and not necessarily being produced as a vehicle for intercellular communication. So that's the next question I want to address, because we've tackled this quite directly, to ask whether we could test in our experimental system the possibility that exosomes could deliver information to a target cell. The reason that this is, I think, been overlooked in the field and assumed to be the normal means by which exosomes work, the problem is that in order for exosomes to deliver their content to a target cell with any efficiency, there would have to be a membrane fusion reaction. By analogy to envelope viruses, which have evolved their own fusogens to do this, exosomes, in order to be effective in doing so, would have to have a fusogen. There are very few fusogens, known fusogens, that act on the extracellular surface. Of course, there are proteins that promote membrane fusion inside the cell, but that's on the other side of the membrane. This would have to be a membrane fusion reaction that was that operated in the exact same in the same manner as an envelope virus. And the question is are there any such fusogens? Indeed, is there any evidence that exosomes can really efficiently deliver their content? So now I'm going to tell you about the experiments of another postdoc in the lab, Zhongyan Zhang, very talented postdoc, who set out to test that possibility by building a vesicle produced in HEC293 cells into which we could efficiently introduce genome editing functions, specifically Cas9 and a guide RNA. We've done so, and I'll show you how we did that. And then we asked, can the vesicles that contain enzymatically active functional Cas9 and a guide RNA, can they find their way into the cytoplasm and then into the nucleus and then elicit a genome editing reaction that we can monitor in real time? So that would require a membrane fusion event. So let's see if that happens. So here's the trick that Zhongyan and I devised for this effort. It's a very simple idea based on another technique that's used to synchronize proteins in the secretory pathway. It's called the rush system, and it's shown here. The idea was to take the gene for, let's see, no, we want to do this. Take the gene for CD63, and instead of fusing N-luciferase to the 3' coding region, to the C terminus, we fuse streptavidin. So now you've got CD63 in the endosome with streptavidin facing the cytoplasm. Correspondingly, we took the gene for Cas9 and fused to its N terminus a protein called streptavidin binding protein. And then we expressed in the donor cell, in this case, the cell is a HEC293 human embryonic kidney cell line. Under these conditions, we expected that the streptavidin binding protein would adhere to streptavidin and carry Cas9 and a guide RNA piggyback into invaginations that bud into the interior of the endosome, allowing a multivesicular body eventually to accumulate vesicles that had an enriched population of Cas9. And then, under conditions that cause these endosomes to fuse at the plasma membrane, we would be able to isolate from the conditioned medium a pool of these vesicles that had Cas9 inside. We were able to do this. We were able to show that the Cas9 remained enzymatically active, and I'll just now show you one experiment that illustrated how we go about doing this. So here's the experiment. So HEC293 cells expressing both fusion cassettes or just Cas9 expressed independently, not in a manner that would allow it to bind and be internalized, were compared in 3 separate samples. The cultured medium was collected and centrifuged to pellet the vesicle fraction. The vesicles were subjected to buoyant density flotation, accumulate at this interface, were collected. The vesicles were shown to be intact. We could show that the vesicles had had Cas9 and the guide RNA inside the vesicle, and we showed the guide RNA was there as well. But then when we examined those vesicles and compared them to vesicles isolated from cells that had just expressed Cas9 alone, not in a manner that would allow it to be tethered to CD63, we found by immunoblot, no Cas9 in the vesicles from these control samples. That is, the vesicles are produced, but there is no concentrative capture of Cas9 into these vesicles. In contrast, when both fusion cassettes are expressed, there was abundant Cas9 packaging into the vesicle. Correspondingly, we could ask whether the guide RNA is packaged by simply exposing the isolated vesicles under these controlled conditions or in the experimental sample to exogenous ribonuclease, and we could show abundant guide RNA packaged into these vesicles, resistant to exogenous ribonuclease unless detergent was added. And this was a substantial enrichment of about 15-fold. Now, on the other side, Zhongyan had to build a reporter that would allow the detection of enzymatically active Cas9 to edit the genome to produce a signal that could be measured easily. And the logic of his construct is shown here. Reduced to its simplest form, he introduced a crippled form of N-luciferase that had a stop codon introduced in the coding frame upstream of the coding sequence of the gene. Not only a stop codon, but a single nucleotide deletion so that this gene could not be expressed unless Cas9 is present, where on introducing a double-strand break, there is an error-prone process of non-homologous end joining that would occasionally restore the reading frame and allow N-luciferase to be expressed. Now that actually works in spite of the complexity of it. If you introduce Cas9 and if you introduce the guide RNA but without Cas9, you don't see any expression of N-luciferase. But if you introduce Cas9 in one of two different guide RNAs, there's a 100-fold increase in the expression of N-luciferase. So that works. This is a reporter cell line that just happens to be, in this case, a tumor cell line, the same MB231 tumor cell line that I described in the first part of my talk. So that works. So we're all set. This was a lot of work just to create the donor acceptor, so we're very excited. But unfortunately, the result that we got then was rather disappointing. In the presence of a 100,000-fold excess of isolated vesicles that we know were internalized by the cell because we could observe these in fluorescence microscopy, 30% of the cells took up these vesicles into an endosomal compartment. A quite moderate 50% increase in the expression of enzymatically active N-luciferase was produced. This in spite of the fact that if the Cas9 and guide RNA are expressed directly in the tumor cell line, there was a 70-fold increase in the expression of N-luciferase. So what happened? What went wrong? Well, there are many possible explanations, but the signal is real. This modest signal is real. It seems to be dose-dependent, as you see here. Well, we did some simple experiments to convince us that we hadn't damaged the vesicles in their isolation, or they didn't have to be consumed right away during the course of the incubation. Here is one such experiment. If you take conditioned medium from the donor cells and you simply add that to the breast cancer cell line without any purification, over the course of 3 days, no increase in the expression of enzymatically active N-luciferase was observed. Even after 8 days, maybe a 2-fold increase, still very modest. But the experiment that really convinced us that there was something fundamentally wrong in the hypothesis is when the two cells, the donor cell, type A, and the acceptor cell were co-cultured in a transwell chamber separated by a membrane-permeable vesicle, such that over the course of 6 days, the vesicles should be able to go back and forth and interact with one another. Even after 6 days, no additional signal was observed. So this was disappointing, but there was one more experiment to do. And that, of course, will be the story that I'll tell you now. When Song Yan took the two cells, the donor and acceptor cell, and cultured them together under conditions where there was a possibility of physical contact, the result was very different than what I've shown you just now. So here's one of the experiments. In this case, HEC293 cells serve as a donor, and one of 4 different tumor cell lines is used as the acceptor, the winner being this cell line MB231, this breast cancer cell line. And the 4 conditions that are shown in each incubation are different conditions. One where the 2 fusion cassettes are present, and one where just Cas9 is expressed by itself, not tethered to a membrane. And even under those conditions, when the acceptor cell line was a tumor cell line, at least a 20-fold increase in active N-luciferase was produced. So that was surprising. But what was even more surprising was, if HEC293 was used both as a donor and as an acceptor, that is, one was constructed to secrete Cas9-containing vesicles, and the other was constructed so as to have a dormant form of N-luciferase. Under those conditions, where the two cell types were allowed to mix together for days, no increase in N-luciferase was observed. So these results suggested to us that there was some asymmetry in this process. A donor cell line, in this case not a tumor cell line, very actively produces something that can be recognized and taken up by an acceptor cell. In this case, an acceptor cell line has to be, at least in our limited experience, a tumor cell line. Now, what could it be? Well, there were many possibilities for maybe a kind of an immune synapse where vesicles would be in a very protected environment, conveyed one to another. Other forms of endocytosis maybe were involved, but we tested all of these alternative views would have required some kind of endocytic process for the transmission of Cas9 from one cell to the next, and those showed no effect of inhibition of endocytosis. So we were left with another hypothesis, namely a phenomenon that had actually been observed over many years, and that is cells in culture, and likely tissues, communicate, we think, by extending membrane tubular connections that can bind to and perhaps occasionally fuse with the target cell. So that would require some kind of a fusogen. So is there such an interaction that explains this rather efficient transmission of Cas9? So these are structures that have been called tunneling nanotubes. Sometimes you can see in cell cultures these structures spreading throughout a layer of cells. They're often very narrow, but they're not exclusively narrow. Sometimes tumor cells produce fat tubules that can appear to touch and connect. What is known about these vesicles is that they can be produced by both donor and acceptors. They may meet. There is certainly evidence that these tubules can project and adhere, but not fuse. But it's also suggested that there must be occasionally some fusion event, because it would require fusion for information to be transmitted between cells. Merely touching a tubule to another cell is not going to be sufficient. It's known that these structures require actin for their formation and stabilization. Indeed, people have seen over the years many things, not only soluble proteins, but aggregates of proteins, aggregates of nucleic acid, even organelles such as endosomes and mitochondria are seen to be transmitted either one way or both ways across these tubular connections. So Song-Yan established a way that we could visualize the formation and possibly the fusion of these structures by using the donor and acceptor cells under conditions where they can be distinguished in the fluorescence microscope. And here's one image. The setup is the donor cells, in this case, HEC293 cells, now are expressing just a lot of GFP in the cytoplasm, so they fluoresce in this green color. They're very easily distinguished. The acceptor cells are marked by the internalization of a fluorescently tagged form of wheat germ agglutinin, which populates both the cell surface and the endolysosomal network within the cell. And on close inspection, over time, Song-Yan observed tubular connections, often tubules projecting from the donor cell, which touch and occasionally fuse with the acceptor cell. So here's a close-up where at the moment of binding, there's clearly some fusion and some material is being transmitted through this apparent open-ended interaction, which we would argue is how, in our case, the Cas9 was being transmitted. Now, how about with the cells where both the donor and acceptor are both HEC293, where they're incubated together for days, and there was no evident transmission of Cas9. Indeed, there are published images of cultures of HEC293 showing these tubular connections in the literature going back years. And yet, when we repeated the experiment under conditions where transmission was not occurring and watched in time, oops, I want to go back, okay. Watched in time, over 20 minutes, 40 minutes, 90 minutes, even a couple hours, the junctions that form here persist. It's almost as though there's a log jam at the point of contact with a broad region of adhesion between donor and acceptor cell, never apparently resolving by any membrane fusion process. So these occasionally are so stable they stay and they just remain bound. And we believe that the difference between the efficient transmission and the inefficient transmission must be the presence of some kind of catalyst of membrane fusion that's expressed in the acceptor cell. So what are the possible membrane fusogens that could participate in this pathway? There's really only one viral-like membrane fusogen in the human genome. It's a protein called syncidin. There are two paralogs, syncidin 1 and 2. But these proteins typically are expressed only in the early embryo. They are expressed in the trophoblast stage, in a pre-implantation embryo, where the cells in the outer layer of the embryo fuse to form a syncytium that gives rise to the placenta. That is known to be the point of maximal expression of the two paralogs of syncidin. Syncidin is is almost certainly of viral origin from tens of millions of years ago, and different vertebrates have enjoyed that kind of an interaction with retroviruses over hundreds of thousands of years. And there's evidence, for instance, in the mouse, that two equivalents called syncidin A and B of different viral origin are responsible for the same early event in embryogenesis. Here is a cartoon that illustrates the overall kind of organization of syncidin and how it's believed to participate in membrane fusion. Syncidin, like other viral membrane fusogens, is present as a single membrane-spanning protein with a short C-terminal domain, a long extracellular domain, and importantly, a cleavage site, a proteolytic cleavage site that is acted upon by the enzyme furin, which is involved in prohormone processing. This furin cleavage releases or produces an N-terminal domain that is of a typical viral fusogen, and the severed domain independently forms a receptor binding domain that is believed to remain bound, possibly by disulfide bonds, to the processed form of furin. These receptor binding domains cause a cell that is exposing this fusogen at the cell surface to interact with cell surface receptors. There are two different receptors that are known, one that acts on syncidin 1 and another that acts on syncidin 2. Once the cells adhere, it's believed that these domains are released, the fusogen is exposed and introduces it protrudes into the opposing bilayer to bring about a rearrangement of the lipids and cause membrane fusion. So, now, why would syncidin be involved in this process? I told you that it's largely only expressed at a very early stage in embryogenesis. But interestingly, I think of substantial interest, tumors upregulate the expression of syncidin. Indeed, it's known that tumor cells can occasionally fuse, and in the literature, there's some evidence that tumors transplanted into an experimental animal may borrow, by extending a tubule from cells surrounding the perimeter of a tumor, may borrow organelles from neighboring cells and hijack them possibly to nourish the tumor. So that was really an interesting possibility. But for our experiments, the simple thing was to ask, could we ablate the expression of, in this case, syncidin 2, and observe a change in the transmission of Cas9. Indeed, we showed that, and let me show you in one of my last slides. When the setup allows us to visualize donor and acceptor cells, where syncidin 2 has been knocked down specifically in the acceptor cell, we observe, as I showed you a moment ago with cultures of HEC293, a persistent interaction at the point of junction that rarely, if ever, resolves to membrane fusion. So here are higher mag views of these examples where the junction persists, in this case for several hours, with no resolution to fusion. So this is really quite interesting. It leaves us with a lot of very interesting prospects. One is possibly tumor cells produce extracellular vesicles that have syncidin, and maybe tumor cells can produce exosomes that actually are able to communicate information to other tissues. Indeed, there's a prominent theory in this field suggesting that primary tumors may send out exosomes that inform tissues to create a pre-metastatic niche into which cells from the primary tumor may migrate to establish metastatic cancer. And if that theory is true, maybe syncidin is involved in that. Syncidin may be expressed in limited other avenues where there is this kind of communication possible. So okay, I'm just about done. This is my last slide. So here's a model with some more details. We've shown a specific role of one of the receptors for, in this case, syncidin 2. We know that it's expressed actually quite widely. It's certainly expressed in the donor cells, HEC23. This interaction produces, in this case, a fusion that permits either endosomes or free Cas9 to diffuse into the target cell. Eventually, because we've attached a nuclear localization signal to Cas9, it can be imported into the nucleus and can engage in some genome editing so as to allow the expression of N-luciferase. We know that actin is required for this process, and we suspect that many other genes are, and so Song-Yan is very busy looking at other genes that may be required for this new interesting pathway. But even more broadly, I'm quite interested in the prospect that syncidin may be an essential component of tumor proliferation, and if that's so, it presents an interesting possible target for therapeutic intervention. Well, let me show you the people who've done the work. These are the important people in the group. Let me start with a former graduate student, Moraima Tomosh-Diaz, who did the buoyant density gradient separations that allow us to characterize the two populations of extracellular vesicles. The team of Justin Williams and Jordan No, who did the experiments that lead to our view that the production of exosomes and extracellular vesicles may simply be a consequence of a normal plasma membrane repair. And now, most recently, the work of a brilliant postdoc, Song-Yan Zhang, who's given us some really interesting future leads of investigation. Thank you for your attention.